In this two-part video series, we're going to talk about human gene therapy. In the first video, you'll learn about how gene therapy is currently being used to treat human genetic diseases. And then in the second video, we'll go over two specific examples of diseases that are currently being treated with gene therapy. By the end of this two-part video sequence, you should be able to define gene therapy, explain the considerations in deciding whether a disease can be treated with gene therapy, explain the difference between in vivo and ex vivo gene therapy, and understand why some diseases are better treated with one approach than the other. You should be able to explain the differences between lentiviral vectors, AAV vectors, and gene editing, and discuss the pros and cons associated with each approach. And then finally, you should be able to design a hypothetical gene editing strategy to correct a human inherited disease. So first, what is gene therapy? Gene therapy is the introduction of DNA into a patient to treat a genetic disease. The most common form of gene therapy involves introducing a wild-type copy of a defective gene using a viral vector. With this approach, you're not actually fixing the mutant gene that's present in the patient's genome. You're just adding a new copy of the gene into the patient's cells. Or using a newer form of gene therapy, you can actually directly edit a defective gene. And with this approach, you are actually correcting the mutant gene that's present in the patient's genome. This strategy is much less common and has only been used recently. So in this lecture, we're going to focus primarily on the first strategy, where a wild-type copy of the defective gene is introduced into the patient. And this is also called gene addition. When talking about gene therapy, it's important to understand that not all diseases can be treated with gene therapy. So, which diseases can be treated with gene therapy? There are a number of important factors that need to be considered when deciding whether a particular disease is a good candidate for gene therapy. And these factors are basically the same ones that we discussed in the previous video on CRISPR, where we talked about editing human embryos. The first consideration is the disease caused by a single gene? Diseases that are caused by mutations in single genes are much easier to correct than diseases that are caused by mutations in multiple genes. The second consideration is how complex the disease-causing mutation is. Diseases that are caused by loss-of-function mutations in single genes can be most easily treated with gene therapy. If the disease results from a gain-of-function mutation or a mutation with a dominant negative phenotype, this is going to be more difficult to treat because it requires gene editing rather than just gene addition. Basically, you have to get rid of the defective gene, not just replace it. So again, the easiest diseases to treat will be those that are caused by loss-of-function mutations in individual genes. Finally, the third consideration is whether other treatment options are available. If there are effective treatments already available, then gene therapy may not be necessary. An important point to mention is that most diseases are not caused by simple mutations in single genes, and many of the diseases that are caused by mutations in single genes are very rare. So at least as of now, it's still the case that most diseases cannot be treated with gene therapy. Now let's go over some of the specifics of how gene therapy works. First, gene therapy treatments can be either in vivo or ex vivo. In vivo means that the treatment is done directly in the patient. In other words, the gene is introduced into the patient's cells while those cells are still in the patient. In contrast, ex vivo means that the cells are first removed from the patient, the gene is then added to those cells in the lab, and then the cells are transferred back into the patient. In vivo gene therapy works well for diseases that are localized to particular tissues. So an example is retinal disease or retinal diseases where the gene is needed only in the retina. For retinal diseases, you can inject the gene directly into retinal cells. However, in vivo gene therapy is not going to work well for genes that are needed more broadly throughout the body. In contrast, ex vivo gene therapy works well for diseases involving cells that can be easily removed from the body, such as immune cells found in the blood or bone marrow. And examples of these diseases include blood diseases, immune deficiencies, and cancer. So for example, you can remove hematopoietic stem cells or T cells, you can add in the required gene, and then you can transfer these cells back into the patient. There are two general approaches to gene therapy, or more specifically, to how mutated genes are restored. I've already mentioned these. Basically, in the first approach, exogenous DNA is introduced into the patient's cells in a viral vector or a plasmid. And with this approach, the defective gene is not corrected, but a wild-type gene is added. This is commonly done using adeno-associated viral vectors or lentiviral vectors. In the second approach, the defective gene is directly edited, and this can be done using CRISPR, which we talked about in a previous video, or using other DNA editing technologies such as zinc finger proteins. This approach is newer and it's still less common than the first approach. However, gene therapy trials involving direct editing are currently ongoing. Now let's discuss each of these approaches in more detail, beginning with lentiviruses. Lentiviral vectors are modified lentiviruses, which are a type of retrovirus. In these vectors, the gene of interest has been added in place of the normal viral genes. And also the viral replication machinery has been removed, so the vector can't reproduce in the cells. Lentiviral vectors can integrate into the genome, as lentiviruses normally do. So basically, the lentiviral vector is engineered to infect cells, integrate into the genomic DNA of the cell, and then express the gene of interest. But it can't reproduce or do any of the other harmful things that a lentivirus normally does. An advantage of lentiviruses is that they can very efficiently target hematopoietic stem cells, T cells, and many other cell types. Also, they are capable of delivering large genes to cells. And they integrate into the genome, so they can be used to target dividing cell populations such as stem cells. If they didn't integrate into the genome, they wouldn't be passed on to daughter cells when a cell divides. So for example, you wouldn't be able to introduce the gene into a stem cell and then have that stem cell give rise to other cells that also express the gene. But since lentiviruses do integrate, they can be introduced into a stem cell or other dividing cell populations. And then all of the cells that are derived from that stem cell will express the gene of interest. However, this can also be a con of lentiviruses because when they integrate into the genome, they can cause gene disruptions. This was a problem with some of the early retroviral vectors that were used for gene therapy. Some of these vectors tended to integrate into the 5' untranslated region of genes. And if this integration occurred upstream of an oncogene, it sometimes resulted in cancer. So the solution used today is to use retroviral vectors, primarily certain lentiviruses, that actually preferentially target coding regions rather than 5' untranslated regions. Now let's talk about adeno-associated viral vectors, or AAV vectors. AAV vectors are modified adeno-associated viruses, or AAVs. And just like with the lentiviral vectors, the genes of interest have been added in place of the normal viral genes. And also, just like with the lentiviral vectors, AAV vectors can't replicate in cells. A really important difference between lentiviral vectors and AAV vectors is that unlike lentiviral vectors, the AAV vectors cannot integrate into the genome. Cells that are infected with AAVs do not pass the virus onto their daughter cells. So basically, AAV vectors can only deliver the gene of interest to the cells that they infect. So what are the pros and cons of AAV vectors? Well, one pro is that they can infect many different cell types. So they can be used to introduce genes into many different cells. Another advantage of AAV vectors is that they do not integrate into the genome, so they are considered safer than lentiviruses, because you don't have to worry about them hopping into the genome and disrupting expression of endogenous genes in the process. However, the flip side of this is that since they do not integrate, they can only be used to target non-dividing cells, or long-lived, terminally differentiated cells. So for example, you can use AAV vectors to target retinal cells, and we'll talk about an example of this in the next video. However, you can't use AAV vectors to target hematopoietic stem cells because the immune cells that are produced from the infected stem cells would not have the AAV vector. Another disadvantage is that AAV vectors can only be used to deliver small genes, since they're not capable of carrying large genes. Okay, finally, the other way to do gene therapy is to use gene editing to modify endogenous genes. In this case, you can use the CRISPR-Cas9 system, or zinc fingers or TALENs, to directly edit the endogenous gene and correct the disease-causing mutation. This can be done either ex vivo or in vivo. If it's done in vivo, then you can use non-integrating viral vectors, like an AAV vector, to deliver, say, Cas9, the guide RNA, and a template for homology-directed repair into the cells of interest. If it's done ex vivo, then you can use either a non-integrating viral vector or you can directly transfect the CRISPR components into the cells of interest. An advantage of gene editing is that you can target dividing cells such as stem cells, while at the same time avoiding the genotoxicity associated with integration of a viral vector. Another advantage is that since you are correcting the endogenous gene, expression of that gene will be controlled by its endogenous promoter. And this is in contrast to gene addition, where the gene typically ends up being expressed at much higher levels than normal. Also, you can use gene editing to correct mutant genes that generate malfunctioning proteins. You can't do this with gene addition, since even if you add a wild-type copy of the gene, the malfunctioning protein will still be there. But if you use gene replacement, you can correct the endogenous gene so you can eliminate the malfunctioning protein. So that's a major advantage of gene editing. Two major disadvantages are that, first, the frequency of genome editing is low in some cell types. And then second, off-target modifications are a major concern. Sometimes you can get double-stranded breaks at unintended locations in the genome, and if this happens, you could be introducing unwanted mutations into the genome. And finally, since gene editing to correct human disease genes is relatively new, the safety risks are still not entirely clear. For example, a recent study found that many people actually have anti-Cas9 antibodies, which raises the possibility that CRISPR-Cas9 gene editing could cause a serious immune reaction in some people. So more research involving gene editing is clearly needed, but there are clinical trials involving gene editing that are currently underway. Now, I just want to give you a brief history of gene therapy. Gene therapy trials have actually been going on for quite a long time. The first gene therapy trial in the US was conducted in 1990 when a 4-year-old with severe immunodeficiency received gene therapy and was successfully treated. Unfortunately, after that, not all gene therapy trials were as successful. There was a major problem in 1999 when a teenage patient died in a gene therapy trial from a massive immune reaction to the therapy. And then in 2002 to 2003, a number of children in a gene therapy trial in France were diagnosed with leukemia resulting from their gene therapy. And this is thought to have happened because the viral vector, which was different from the ones that are currently used today, the viral vector integrated upstream of an oncogene, and this caused the oncogene to be overexpressed, which resulted in cancer. So from these cases, it became clear that more research was needed before gene therapy could be considered safe. And so over the next few years, scientists developed much safer methods for gene delivery, such as the use of AAV vectors and the lentiviral vectors that are used today. But around the same time, there also were some successes. For example, in 2003, a gene therapy for head and neck cancer was approved in China. And then a few years later, in 2007, gene therapy was successfully used to treat a vision disorder in Britain. And then beginning in 2010, there have been many successful gene therapy trials as a result of improved gene delivery methods. 2017 was a particularly exciting year for gene therapy in the United States because the FDA approved 2 different gene therapies. One is CAR T-cell gene therapy, which is being used to treat leukemia, and the other is called Luxturna, which is being used to treat an inherited form of blindness. This table shows gene therapies that are either approved or in clinical trials, and you can see that there are a lot of them that are currently being successfully used to treat patients with otherwise intractable genetic diseases. So this table shows ex vivo gene therapies. One of these is CAR T-cell therapy, which you're going to learn more about in discussion section. And another is a gene therapy to treat ADA-SCID, an immune disease that we'll discuss in the next video. And this table shows in vivo gene therapies that are approved or in clinical trials. One of these is a therapy to treat spinal muscular atrophy, or SMA, which you'll learn about in class. And then another is the Luxturna therapy for inherited blindness, which you'll learn about in the next video. So hopefully by the end of this segment on gene therapy, you'll have a new appreciation for the wide range of genetic diseases that are currently being treated using gene therapy.